Welcome back everyone to this third and final component of the urinary system. And in this part of the topic, we're going to learn about how the urine is concentrated. We're also going to talk about hormonal regulation of urine volume and concentration. And then we'll finish up by talking about other components or parts of the urinary system. So let's start with this urine concentration mechanism, which is a mechanism that aims to maintain fluid balance in the body, so in the blood. Now, when large volumes of water have been consumed, your kidneys have the ability to eliminate that excess water without losing large amounts of valuable electrolytes in that high volume of urine. So the response is that the kidneys produce a large volume of dilute urine. Okay, the opposite then happens when drinking water is not available. So the kidneys produce a small volume of concentrated urine. And this still allows us to be able to remove waste, so all of those wastes, but because we're only producing a small volume of concentrated urine, it's going to prevent rapid dehydration. So these mechanisms that create urine of variable concentration rely on a few different factors. Okay, if you look over here at this diagram, it's just showing you a nephron. We've got our glomerulus here where we have glomerular filtration. Then we have the proximal convoluted tubule where most of that reabsorption is occurring. And remember, there is secretion in there too. Then we go down into this descending part of the loop of Henle, and it has it is permeable to water, right? It has these little water channels called aquaporins that allow water to pass from the filtrate into the interstitial fluid. Okay, then we have our ascending limb of the loop of Henle that is impermeable to water, right? So the volume of water in the filtrate here is not going to change because the water can't get out, but it is permeable to solutes. So those solutes can be pumped from the filtrate out into the interstitial fluid, right? Then we have the distal convoluted tubule here, which selectively secretes and absorbs different ions to maintain blood pH and electrolyte balance, right? And then finally, we have the collecting duct, which reabsorbs solutes and water from the filtrate. And here I'm really going to be talking about antidiuretic hormone here, and we'll also talk about aldosterone as well in terms of the control that they have on this distal part of the nephron in determining how concentrated the urine is going to be. Now, what I need you to get into your heads, right? This is very complex, right? And we're not going to go into it in too much detail. But as you descend down into the inner regions of the kidney, so through the cortex down deep into the medulla, you're going to get an increased salinity. Okay, and by the time you get to kind of the U-bend of the loop of Henle, it is really, really salty. Okay, so it's 1200 milliosmoles per liter compared to up in this cortical region and the blood plasma in particular, where it's only about 300 milliosmolar. And so this amazing, like I am blown away every time I, sort of think about this mechanism that we have that concentrates our urine, because it allows us to maintain our blood osmolarity of about 300 milliosmoles per liter, regardless, of whether we've taken on lots of water or not. Okay, now it is dependent on a few different factors, right? The first one is the maintenance of a high concentration of solutes in the medulla. So be thinking sodium chloride here. Countercurrent functions of the loops of Henle. Now, what does that mean? Well, a countercurrent function means, you've got two sets of parallel pipes. So you can think of the descending loop of Henle and the ascending loop of Henle as kind of countercurrent loops, right? So you've got fluid flowing in opposite directions through those two different tubes. So obviously the filtrate will flow down this way through the descending loop of Henle and then back up in the opposite direction towards the cortex there in the ascending loop of Henle. And then also the control of the permeability of the distal nephron to water via those hormones that we're going to talk about, okay? And so all of those things, will allow us to produce urine of variable concentration. So looking a little bit closer at this medullary concentration gradient that does exist. And without it, we wouldn't have the ability to concentrate our urine. In order to concentrate urine and prevent a loss of a large volume of water, the kidney has to maintain this high concentration of solutes in the medulla. Now, as I mentioned on the previous slide, the interstitial fluid concentration is 300 in the cortical regions and the outer region of the kidneys, and then gradually it increases to 1200 at the tip of the pyramids in the medulla. Now, the maintenance of this gradient itself depends on those functions of the loops of Henle, right? So those sections of the loops of Henle that are, permeable to water, so the descending limb of loop of Henle, and then the ascending loop of Henle being impermeable to water but permeable to solutes. Also, while you have a countercurrent mechanism of the filtrate running through the loops of Henle, you also have the vasa recta. Remember those straight blood vessels that course right down following the loops of Henle into the medulla, and they flow in a countercurrent way to the filtrate in the loop of Henle. And I'll show you a video on that in a minute. And also the distribution and recycling of urea also adds to this concentrated medullary interstitium, okay? And so this high solute concentration in the medulla is created and it's maintained via the following mechanisms. And I have kind of mentioned some of them. Active transport of sodium, followed by co-transport of ions such as potassium and chloride and other ions out of this thick ascending limb into the interstitium. So pumping those solutes out, right? Impermeability of the ascending limb of the loop of Henle to water, okay? So water cannot get out here, solutes can get out, right? And as solutes are pumped out, that means that, the filtrate flowing back up to the cortical region is going to get more dilute, okay? But it's going to stay kind of, really concentrated down here. Okay, we have the vasa recta, have the ability to remove excess water and solutes that enter the medulla without destroying the high concentration of solutes in the interstitial fluid of the medulla. We have active transport of ions from the collecting duct into the interstitial fluid of the medulla, right? And passive diffusion of urea from the collecting duct into the interstitial fluid of the medulla, and then those structural areas of the loops of Henle, so the impermeability of the ascending limb and the permeability of the descending limb of the loops of Henle to urea, all create and maintain that high solute concentration in the medulla, which allows us to concentrate our urine. And so let's see how it all works now, and this will make it much clearer, I hope, for you, apart from all of those, different sort of mechanisms I've been talking about. Firstly, here's our loop of Henle, okay? And we've got our glomerulus here with filtration occurring, and our filtrate then flows down the descending limb of the loop of Henle, turns the corner, and then flows up into the ascending limb of the loop of Henle, into the distal tubule, and then through the collecting duct, okay? Now, what I want you to, and then in red here, we've got our vasa recta that you can see coursing down, following the limbs of the loop of Henle. But you can see straight away that the filtrate here is flowing downwards in this direction, upwards in that direction. That's your first countercurrent mechanism, right? But also, if you look here, we've got filtrate flowing this way, and we've got blood flowing in the opposite direction there, and it's occurring on both sides, right? So again, that's a countercurrent mechanism too. All right, so what I'm going to overlay now here is looking at this thick ascending limb of the loop of Henle, which pumps out sodium and is impermeable to water. Okay, so sodium enters the vasa recta, and that will make the blood in the vasa recta hyperosmotic. So it's becoming more salty because of all these solutes that are getting pumped out as that blood heads down towards the deepest part of the medulla. Okay, so let's move to the next slide, keeping in mind, that the salt is carried down into the medulla of the kidney, making this region hyperosmotic. If we now look at the thin descending loop of Henle, it's permeable to water. That's just a structural thing, okay? So water leaves. This is so amazing, right? Water is going to leave via osmosis, right? Why is that? Because this, medullary interstitium is really concentrated, so there's a gradient there that's going to pull that water out, and it's going to be carried away by the vasa recta. So this also helps to make the medulla hyperosmotic. Now then, if we go over to the collecting duct, as the collecting duct passes through the hyperosmotic medulla, water is going to try to leave, right? Because this is really, really salty, and water wants to get out to dilute out all of that salt. But this is regulated by antidiuretic hormone. So if you've taken in lots and lots of water, antidiuretic hormone is not released. So obviously then, that water will just pass through and you'll excrete high volumes of dilute urine. But if you're a bit dehydrated, antidiuretic hormone is released from the posterior pituitary, and it comes along and it binds to its receptors on cells lining the collecting ducts to cause the insertion of these little pores called aquaporins, and then water is going to be freely reabsorbed because of this concentrated interstitium. Like, if this doesn't blow your mind, about how our kidneys are functioning to regulate our fluid and electrolyte balance by, allowing us to concentrate urine when need be, I don't know what else will sort of amaze you, because I find this fascinating. And this slide here just summarizes it all for you. You can see the cortical region, and then as we descend down into the medulla, we're getting more and more concentrated, right? And then this will just show you how the osmolality of the filtrate kind of changes, depending on, well, obviously, as we descend down further into the interstitium and then back up it towards the distal convoluted tubule. Now, because of everything that I've been talking about, what I need you to sort of understand here is this filtrate that's about to enter the distal convoluted tubule is very dilute. So it's hypoosmotic to blood osmolarity. And that really sets up the scene for then, that concentration to occur via antidiuretic. And so just to summarize the mechanism of action of antidiuretic hormone, which we've been through in several topics now, when it's released from the posterior pituitary gland, so when somebody, has high blood osmolarity or is dehydrated or, low blood pressure, it travels all the way down to the cells of the collecting duct, where it binds to receptors on those cells to cause the insertion of these aquaporin pores into the cell membrane, and that then will increase the flow of water out of the tubule. And the reason, and I keep repeating myself, the reason you can have that increased flow of water out of the tubule cell to be recovered by the blood is because of that concentrated interstitium that's present. And this will further clarify it for you here. In the presence of antidiuretic hormone, the collecting duct becomes permeable to water. Water is reabsorbed then into the interstitial fluid via osmosis, and then it'll be picked up by the vasa recta. And so the result then is the production of a small volume of concentrated urine. When you're really hydrated, the collecting duct doesn't, have the, effect of antidiuretic hormone because it's not released from the posterior pituitary gland. So the collecting duct stays impermeable to water, and more water remains in the collecting duct. And then the result then is the production of a large volume of dilute urine. So overall, antidiuretic hormone influences water reabsorption. It reduces water loss by increasing the number of pumps returning water to the body and to the blood. Of course, other hormones that regulate urine volume include aldosterone and atrial natriuretic peptide. And these are those two antagonistic hormones that have opposite effects on sodium reabsorption. So as water is reabsorbed, the total volume of urine is reduced by the amount of water removed by the tubules, right? Aldosterone is that steroid hormone secreted via or by the adrenal cortex. It increases the distal tubule reabsorption of sodium, raising the sodium concentration of the blood and therefore promoting reabsorption of water. Atrial natriuretic peptide, on the other hand, then is secreted by atrial muscle fibers, and its action opposes aldosterone because it causes the kidneys to reabsorb less sodium and therefore less water. And so the net effect of atrial natriuretic peptide is to produce increased volumes of urine. So aldosterone is known as a salt-retaining hormone. It's a steroid secreted via the cells of the adrenal cortex when blood sodium concentrations fall or when potassium concentrations increase. Remember as well that it's also secreted via the renin-angiotensin-aldosterone mechanism, where we have, that drop in blood pressure causes renin to be released. Renin then, catalyzes the conversion of angiotensinogen to angiotensin 1, which is rapidly converted to angiotensin 2 by angiotensin converting enzyme. And then that then stimulates the adrenal cortex to release aldosterone. It acts on the thick segment of the nephron loop, the distal convoluted tubule, and the cortical portion of the collecting duct. It promotes sodium reabsorption and at the same time will, cause increased potassium secretion. Water and chloride follow the sodium ion, and the net effect then is that the body retains salt and water, which helps to maintain blood volume and blood pressure. Atrial natriuretic peptide then is secreted by atrial myocardium in response to high blood pressure. It has several actions that result in the excretion of more salt and water in the urine, and therefore, if you've got higher sort of volumes of urine, that's going to reduce the blood volume and the blood pressure. So how does it do this? It dilates the afferent arteriole and constricts the efferent arteriole. And remember how we talked about that would then cause increased glomerular filtration rate and therefore increased formation of filtrate. It inhibits sodium chloride reabsorption by the collecting duct. It also inhibits renin and aldosterone secretion and also antidiuretic hormone secretion. So the net effect of atrial natriuretic peptide is to increase sodium excretion and therefore water excretion and therefore higher volumes of urine excreted to reduce blood volume and blood pressure. And this is just a summary slide, showing this regulation of urine volume in terms of antidiuretic hormone. I'm not going to repeat all this again. Aldosterone and also atrial natriuretic peptide, and those little circles there indicate where aldosterone and AMP are acting, and obviously this blue arrow here indicating water reabsorption from the collecting duct. And finally, looking at the effect of parathyroid hormone on kidney function. Remember about parathyroid hormone from topic 1. It's secreted from parathyroid glands and responds to lower than normal levels of calcium in the blood, so hypocalcemia. Its net effect, and when it's in excessive levels particularly, is that it acts on the proximal convoluted tubule to increase phosphate excretion. But at the same time, it acts on the thick segment of the ascending limb of the loop of Henle and on the distal convoluted tubule to increase calcium reabsorption from the filtrate. So overall, the net effect of parathyroid hormone is to increase the phosphate content of the urine and lower the calcium content in the urine. And here's a true-false activity for you to do to see if you understand how the kidneys can concentrate urine. You'll also find a link to this one underneath the recording on the Brightspace site.